All right, guys, we are going to be getting into our next unit, which is decoding your future. We're going to start talking about DNA, RNA, transcription, translation. There are so many things that go into it, and it's such an amazing process. And this is definitely one of my favorite things that we are going to learn about in biology. Now, just something for you to consider. Our topic question is, how does the structure of DNA determine proteins? So by the end of this, I want you to be able to describe the structure of a DNA molecule. And here is your warm-up question that you can answer or just kind of be thinking about in the back of your head. Which of these organ systems is responsible for the removal of metabolic waste from the blood? Is it going to be your endocrine system, your nervous system, your muscular system, or your excretory system? All right, now let's get into the structure of DNA. Now, I have a list of 2 roles of nucleic acids, which are going to be your DNA and your RNA. Their main job is to store genetic information, direct the assembly of proteins. Now, what are the subunits of DNA and what does each subunit contain? So the subunits of DNA, which is what builds it up, is going to be your nucleotides. So these subunits will contain your sugar, which is deoxyribose. That's where the D in DNA come from. It's from deoxyribose, which is the sugar that we use to build DNA. You also have your phosphate group, and you have your nitrogenous bases, which are going to be A, T, C, and G, which also stand for adenine, thymine, cytosine, and guanine. Now, if you'll notice the little bracket I have here off of our sugar and phosphate group, that is always going to be the same with your DNA molecules, so just keep that in mind. So here is a DNA structure. So we have our blue pentagon shape, which is representing our deoxyribose sugar. We have our base. So this is where we're going to find our nitrogenous bases of adenine, thymine, cytosine, or guanine. Then over here is where you're going to find your phosphate group. Now, let's get into complementary base pairings. If you've never heard of Chargaff's rule, you're going to learn a lot about it. So this was established in 1949. Chargaff's rule states that in a DNA molecule, the amount of cytosine always equals the amount of guanine. So C is always going to equal G. And the amount of adenine bases always equals the amount of thymine, which is A to T. Now, the double helix shape was determined by James Watson and Francis Crick, with also the help of Rosalind Franklin. Of DNA, our double helix shape of DNA is often going to be compared to a spiral staircase. So if you notice my pictures here in the background, you can see that it's twisted and you have these little strips that go across. These little strips are going to be your base pairs, your cytosine, guanine, adenine, and thymine. So if it was just straight up and down, it kind of looked like a ladder or a staircase, but that's why we refer to it as a spiral staircase, because you notice that there are twists throughout the molecule. Now, keeping Chargaff's rule in mind is very, very important, because cytosine is always going to equal guanine and adenine is always going to equal thymine. That's really important to keep straight, so I want you to keep that in mind. Now, here are some more pictures for you to get a better idea. And And you'll also notice we have our twisted staircase to show you. And here's a comparison so you can really get a better idea of what we're talking about. And here we have a better representation of our complementary base pairs and how they'll pair up together. So if you notice, it doesn't really matter which one is to the left or the right, because C and C can be on the left or the right, and same thing for a T and a T. So it doesn't matter which side of the molecule it's on. A is always going to pair with T, and C is always going to pair with G when we are referencing our DNA molecule. Now, here is our picture. This is Watson and Crick. So these are the guys who we credit the founding of DNA with. Now, this is Rosalind Franklin. She actually did a lot of the footwork for the discovery of DNA. Franklin did a lot of the research and early development of DNA, but because of the time that this was taking place in, which was the 1940s, women were not taken seriously in most aspects of daily life. So Watson and Crick actually kind of stole her research to take advantage of her findings. So even though we do say that Watson and Crick are the fathers of DNA, I feel like Franklin does not get the credit she deserves for her early work in DNA. And if you'll notice here, this is an early picture of what we thought DNA looked like. So let's talk about complementary base pairing again. So the sides are going to be composed of the sugar and phosphate groups. So the rungs are composed of the bases. The bases are joined together by weak hydrogen bonds. Cytosine always bonds with guanine, and adenine will always bond with thymine, just like we talked about a few slides ago with Chargaff's rule. So a mnemonic device that I like to share that is super funny, but it helps you keep it straight. Cool giraffes are tall. Oh my gosh, we all knew that giraffes are tall, but cool giraffes are tall. And if you notice, the first letter of each one of our words is cytosine and guanine, adenine and thymine. So you can keep those base pairings straight. And if you cannot remember the names, you can always remember the letters. If you remember that cool giraffes are tall. Cool giraffes are always tall, guys. Now, here is a picture for us to look at. So we have our DNA molecule as a whole, but if we magnify this part of it, which are the stairs of our staircase, that's where it's going to show us our sugar phosphate backbone, which is going to be here to the side, because we have our phosphate molecule and then we have the building here. Because if you look a little bit more closely, you'll notice our pentagon shape here. Oh my gosh, we remember that pentagon shape. That is our deoxyribose attached to our phosphate group. And here, this is where you're going to have your nitrogenous base pairs. So it's going to be your C, your G, your A, or your T. And the thing that's going to hold it together on each side, because think about this as a mirror per se. It's showing the opposite of what is on this side. And the thing that connects them is those weak hydrogen bonds. So here's a little diagram for you, and I have included this on your notes. I want you to take a minute to fill out this 2D diagram. I want you to tell me what this diagram is, and I want you to label each part of the DNA molecule, and I want you to give me the complementary base pairs. And I will give you a few minutes to work on that. Alright, guys, I hope you didn't struggle too much with that. So let's move forward. So this is a 2D structure of DNA. Out here is going to be your phosphate group. These are going to be your deoxyribose sugars. So here, attaching our base pairs is going to be your hydrogen bonds. And this as a whole is going to be your nucleotides. And I hope you got all your base pairs correct. Remember, cool giraffe starts all. Your adenine is going to pair with your thymine, just like thymine is going to pair with adenine. And then your cytosine is going to form with guanine. Now here is a beautiful 3D diagram of DNA. So you can see all the phosphate groups that are going along through here and the stairs. And over here is a better picture to show you everything that goes into connecting your DNA together. So I've added a lot of visual representation here so you can get an idea of where all this is coming from, because you're going to see so many different types of models depending on what we're looking at and what we're studying. So I want to give you a good idea of all the different ways that you could possibly see it, because I don't want you to be caught by surprise. Now, let's talk about eukaryotic DNA replication. So, if you recall back to learning about the cell and its organelles, all the DNA is stored in the nucleus. So it shouldn't surprise you that DNA replication will occur in the cell's nucleus during the S phase of interphase of the cell cycle. Now, we're going to talk about interphase and all the different parts of the cell cycle later on. But I want you to know right now that DNA replication is going to happen in the S phase of interphase. Now, at the end of DNA replication, each chromosome will consist of two sister chromatids connected at the center by a centromere. So if you'll notice, it kind of just looks like an S with a dot in the middle. So one piece of the X is one chromatid. So that's why we call it sister chromatids, because they're stuck together, connected by this centromere. So it kind of just looks like an X. So let's talk about DNA replication. And I've broken this down into steps for you so you can more easily understand the process. So step 1 of DNA replication. The DNA double helix is going to be unwound and unzipped by an enzyme called helicase. So think about our twisted DNA molecule. The molecule is going to untwist to be able to straighten out. And just imagine like you're unzipping your jacket. Your base pairs are going to separate through the DNA molecule to make two separate strands, as the enzyme helicase acts as your zipper to separate it. Now, the sites where separation occurs, which sites is just where we see the separation happening. So where the sites separate and replication occurs, we call that the replication forks. Just like you're coming up to a fork on a road and you have to make a choice whether to go left or right. So you're sitting there at the crossroads. That is your replication forks. And DNA replications occurs at hundreds of places in the DNA. Now, step 2. Each of the two strands serves as a new template for the addition of new nucleotides by the enzyme DNA polymerase. So DNA polymerase adds nucleotides according to Chargaff's rules. So imagine that we are unzipping the DNA molecule, but then we're going to be making new DNA molecules with Chargaff's rule. So here is a good picture for you to be able to actually see it. So we start off with our original DNA molecule here. So this is the DNA helicase, and you'll see here your replication fork. So your helicase is kind of going to be like a little bubble here. So your bubble is moving up the DNA molecule, and it's causing it to separate, just like you would if you were unzipping your jacket when you got home from school. So the DNA polymerase will add complementary nucleotides to each strand. Now, when we say complementary nucleotides, that is just our complementary base pairs. So it is different in the strand colors, if you notice. Our new DNA is going to be this new green strand you see coming in, while our old DNA is going to be represented by the blue. So our DNA polymerase is going to work with our new DNA to bring it in and wrap around to create two DNA molecules instead of one. So we have our new DNA and our old DNA, which are paired together to make two identical DNA molecules to the original DNA molecule. Now, the parent molecule has two complementary strands of DNA. So each base is paired by hydrogen bonding with its specific partner, A with T or G with C. So the first step in replication is separation of those two DNA strands. So we're breaking those weak hydrogen bonds that are holding our complementary base pairs together. So each parental strand now serves as a template that determines the order of nucleotides along a complementary base strand. So our dark blue is going to be our parent templates, and we're going to have our new strands coming in to complementary match our parent strands. So the nucleotides are going to then be connected to form the sugar phosphate backbones of the new strands. So each daughter DNA molecule consists of one parental strand and one DNA strand. So we call it a daughter molecule because our figure over here at A, this is the parent. This is all the parent has. And as we go through this process, think about it like this. Your parents give you pieces of themselves to make you. So this are the pieces your parents give you, and this is what you have. So it's the daughter strand of what's being made by the parent molecule. Now let's talk about semi-conservative replication. This is referred to as semi-conservative replication, in which two identical molecules of DNA are formed after replication. In each DNA molecule, half is new and half is old. So that was kind of represented on our last figure with our old DNA and our new DNA. So this is the semi-conservative replication. So here's an even simpler way to look at it. It's semi-conservative because if you notice, only half of our new DNA strands are going to be new. So your DNA doesn't have to work nearly as hard just making new DNA molecules from scratch. I apologize. They are working to put new bases onto it to add more and more DNA through replication with our new bases. Now, with anything, DNA replication can have mistakes. Because I'm sure you can imagine that things can get messed up in the unzipping process. I'm sure you've been trying to unzip a jacket and your zipper's gotten jammed or something gets caught in it. So let's read about some of these mistakes that can occur. So in bacteria, DNA polymerase will proofread newly attached nucleotides to make sure that they are correct. If a mistake is found, the DNA polymerase will replace the wrong nucleotide. So when I talk about a mistake being found, let's say you're looking at cytosine, guanine should be the one that pairs with it. But an example of a mistake could be that instead of a C and a G being bonded together, a C is trying to bond with a T. So a cytosine is trying to bond with a thymine, which is a problem. That cannot happen. So your DNA polymerase is going to replace the wrong nucleotide. But just like anything else, it can make mistakes too. So if the DNA polymerase misses a mistake, other mismatched enzymes will fix the mistakes. So repair mechanisms in eukaryotes are not truly understood because we have all these things that can run in and repair it, but it doesn't always get repaired. But we don't really know a whole lot about why this occurs or how these mechanisms are in place. Now, causes of DNA replication mistakes. Sunlight can cause DNA replication mistakes because the sun can kill parts of your DNA and actually cause different abnormalities to happen. So that's how you can end up with skin cancer, for example. Cigarette smoke can cause mistakes in DNA replication. Your age can cause mistakes in DNA replication. As you get older, your body's not working as well. So things can happen. And also, mutagens. Now, mutagens is radiation or chemicals that will cause mutations. Think about Spider-Man, for example. Spider-Man was bitten by a radioactive spider, and it changed his DNA to make him Spider-Man. Because you and I cannot just shoot spider webs out of our wrists like the original Spider-Man can. But if we were all bitten by a radioactive spider to change our DNA, we could. So that's just a silly example of a mutagen that can cause DNA replication mistakes. Now, for DNA mutations, if an error is not repaired, that is when the mutation occurs. So, a mutation is any sequence of nucleotides in a DNA molecule that does not match the original DNA molecule when copied. And I'm sure you can imagine what an issue that this could cause, because obviously our DNA is made the way it's made to keep us healthy and to keep all of our information in a organized, fashioned order. But if mutations get in there, it can cause a lot of problems for you and your body. All right, well, that is going to be part 1 of Decoding Our Future. You will be getting a part 2 soon enough. I hope you enjoyed this, and if you have any questions, just be sure